I'd like to thank the organizers today for allowing me to present. I have no disclosures and would like to thank the PA for funding this research. Stress urinary incontinence is the involuntary leakage of urine due to an increase in abdominal pressure. The primary risk factor is childbirth, as it injures the pudendal nerve as well as the external urethral sphincter. Postpartum incontinent women have been shown to have an increased pudendal nerve motor latency, suggesting pudendal nerve recovery and function are important to the continence mechanism. The field of neuroscience has shown that electrically stimulating an injured nerve accelerates recovery as well as enhances BDNF expression, or brain-derived neurotrophic factor. We have shown that electrically stimulating the pudendal nerve 4 times a week for 2 weeks accelerates functional recovery of leak point pressure compared to sham stimulation. If BDNF is important to the accelerated recovery, how can we inhibit it? We can use a previously used tyrosine kinase B, the receptor for BDNF, functional chimera, to bind the free BDNF. This would then inhibit BDNF from binding to the TrkB receptors on the growth cone of the injured neurons. We have previously shown that using this TrkB, we can significantly decrease the amount of free BDNF at 3 weeks after a pudendal nerve injury. Additionally, we can significantly decrease pudendal nerve motor branch firing rate recovery compared to a PNC plus saline treatment. So to our hypothesis that BDNF is important to the accelerated recovery via pudendal nerve electrical stimulation after a simulated childbirth injury. We had 4 groups. We had a sham dual injury that had no electrodes implanted and received a saline treatment. A dual injury, which is the pudendal nerve crush, where the pudendal nerve is crushed bilaterally twice for 30 seconds, followed by a 4-hour vaginal distension, which had electrodes implanted but did not receive electrical stimulation and saline treatment. Then a dual injury that received electrical stimulation with saline treatment, and then a dual injury plus electrical stimulation plus the TrkB. For 2 weeks, the animals either received electrical stimulation, sham electrical stimulation, or isoflurane for 1 hour 4 times a week for the 2 weeks. After treatment had stopped, we waited another 2 weeks, and then we did functional testing, which consisted of leak point pressure, EUS, EMG, and we then harvested the EUS and pudendal nerves. The EUS were stained for neuromuscular junctions, and the pudendal nerves were sectioned and stained for nerve morphology. Here you can see we demonstrated the model as there was a significant decrease in the SUI group with sham treatment compared to the sham injury group. Electrical stimulation then accelerated leak point pressure recovery, while the addition of the TrkB decreased this accelerated recovery. Similarly, in EUS EMG firing rate, we saw that the dual injury or SUI with sham treatment was significantly decreased compared to the sham injury. Electrical stimulation accelerated the recovery of the firing rate, while the addition of the TrkB molecule inhibited this recovery. In the sham injury group, we can see the sensory motor branches, axons, are organized and compact. In the SUI group, we see that they are not organized and not compact. With electrical stimulation, we see that the axons are organized and compact. And with the addition of the TrkB after electrical stimulation, we see that the nerve axons are not as compact or as organized in the electrical stimulation group alone. We then looked at neuromuscular junction staining and saw in the sham injury group compact neuromuscular junctions that were innervated. In the SUI group, we saw non-compact neuromuscular junctions that were not innervated. In the SUI plus electrical stimulation group, we saw compact neuromuscular junctions that were nearly reinnervated. And in the SUI plus electrical stimulation plus track B, we saw non-compact neuromuscular junctions that were not reinnervated. So we showed the track B molecule binds free BDNF. BDNF is important to pudendal nerve functional recovery. BDNF is needed for the accelerated functional recovery after electrical stimulation. And these results suggest that BDNF is important to the recovery of continence after the childbirth injury. I'd like to thank the VA for funding, and if you have any questions, please contact Margot Damaser. Thank you.